The overall goal of this procedure is to isolate SecM-arrested 70S ribosome-bound nascent chain complexes, or RNCs, produced during translation in an in vitro cell-free system. This is accomplished by first introducing a 17 amino acid long SecM stalling sequence to the C-terminal end of the gene of interest and cloning it downstream of the T7 transcriptional promoter. Next, the mRNA is transcribed using an in vitro transcription reaction and then purified. Then, the purified mRNA is translated in an S30 E. coli extract system with radioactive amino acids to label the synthesized protein or nascent polypeptide. Finally, the SecM-arrested ribosome-bound nascent chain complexes are isolated by sucrose gradient centrifugation and fractionation. Ultimately, results can be obtained showing that nascent chains are stably bound to the 70S ribosome by gel electrophoresis analysis of the labeled polypeptides isolated from the gradient fractions.
The main advantage of this technique over existing methods like the use of mRNA lacking stop codon is that it ensures with high efficiency the attachment of nascent polypeptide chain to 70S ribosome during translation and subsequent centrifugation step. Additionally, in contrast to no stop codon technology, this method can be used with almost equal efficiency during both in vitro and in vivo system.
This method can help answer key questions in the field of co-translational protein folding, such as how early during the translation various co-translation folding intermediates might form and further help